Hello. I'd like to thank the organizers for giving me the opportunity to speak here at the Keystone Conference. I'm going to be talking about work that I've done on repeated immunization. Now, the motivation for this is that there's a great deal of recent work on HIV vaccine development that has focused on repeated immunization. These immunization protocols are an attempt to elicit broadly neutralizing antibodies, and the idea is to use multiple different immunogens in series. Now, the one thing that concerns me about this plan is that we don't really understand how multiple vaccines behave in the first place, much less with variable immunogens over time. So I'm going to tell you about the work that we have done in the quantitative characterization of the population dynamics of B-cell clones. Of course, it's the B-cell clones that are doing all the work for the vaccine. And I'm going to talk about both the intraclonal evolution, the affinity maturation, and the interclonal competition, which is elicited by repeated administration of a single immunogen over the course of 18 months. Now, the only drawback of these studies, as far as you're concerned, is that they don't involve HIV vaccine. But the advantage that we have for not using HIV is that we are able to do it in humans. So let's just look at a simplified version of B-cell population dynamics, and I'll try to make clear what it is that we're trying to understand. So there is a pool of naive B cells, and a relatively small number of those will be specific for the immunogen. And when the vaccine is delivered, those antigen-specific B cells will be activated. Some of them will become plasmablasts and eventually plasmacytes, which are the effector cells of immunity, of course. And some of them will go to the germinal center. And in the germinal center, they will experience somatic hypermutation, leading to affinity maturation. And some of those will become long-lived plasma cells, and others will go on to become memory cells. And so after the first immunization, we expect that there will be a pool of memory cells. Now, after the second immunization, the memory cells, which now exist, are presumed to re-enter the germinal center reaction, in part, and some of them to go on to differentiate into long-lived plasmacytes. So the model that we carry around in our minds is that of immunization continually eliciting memory B cells back into the germinal center reaction and increasing their affinity for the immunogen time after time. And we expect then that there will be a relatively small number of clones that persist after multiple immunizations as they outcompete the other clones by simply having a better head start or having a better path to affinity maturation. What we'll find is that is not the case in the studies that we've done. We're going to focus on the plasmablasts because of the technical limitations that we had at the time on the kinds of B cells that we could perform a paired heavy chain light chain sequencing. So the study is going to involve the use of anthrax vaccine adsorbed. And this was chosen as our immunogen because it's FDA approved, and it would be very straightforward to recruit volunteers and to study them over the 5 immunizations that it takes according to the standard protocol. The AVA consists primarily of protective antigen from Bacillus anthracis, although there are small amounts of other proteins in there as well. The PA protein is an 83 kilodalton protein, and it comes packaged with alum. So the experimental design is that we recruit 6 volunteers, and the volunteers each receive a vaccine and are followed up at weeks 1 and 2 post-vaccine with blood draws. So for every vaccine, they get they receive, of course, the vaccine, and they get 3 blood draws on weeks 0, 1, and 2. We are going to focus on the blood draws taken at week 1, at the point where we expect plasmablasts to be in greatest numbers in the peripheral blood. So our strategy is going to be to examine the somatic genetics. We're going to partition all of our sequences up into clones and then follow the behavior of the clones as they grow and diminish and increase their affinity through affinity maturation. And for this, we're using the gating shown on this slide for plasmablast sorting. And we're going to use Atreca's proprietary technology to extract or to isolate the heavy chains and light chains in pairs from individual B cells. Well, we carried out this procedure and obtained just about 10,000 or a little bit more than 10,000 paired sequences representing 10,000 antibodies, and they are distributed fairly evenly across immunizations and subjects. So the first results that we got prior to actually looking at the individual clones are shown in this slide. The upper left component shows serum titers, anti-PA IgG positive serum titers to be precise, over time and with immunizations shown as vertical lines. And you can see that the titers rise until about the 3rd immunization, where they remain relatively flat after that. In B, we're looking at the plasmablasts isolated as a proportion of total B cells isolated. And so they grow consistently up to the 3rd immunization and then shrink again after the 4th immunization. And we're not quite sure what to make of this, but we do think that it is an actual biological phenomenon and not an artifact of the flow sorting, because these samples were not all sorted on the same day. And it might, of course, be the fact that we're using a different lot of the AVA at different immunization points, and that may be a possible source of that kind of change. In C, we're looking at the mutation frequency over time, and this was the first indication that we had something unusual going on. You see that for all of our subjects, the mutation frequency starts high and decreases. So between the 1st and 2nd immunizations, we see a substantial decrease in the mutation frequency. And then from that point on, a much more gradual increase to a plateau by the end of the study. So that's the first phenomenon of interest. If we look at the mutation frequency in a different way, looking at the cumulative mutation frequency, so mutation frequency is now shown on the x-axis, and the y-axis is the proportion of cells with that mutation frequency, in this case in the heavy chain. And the one thing you see is that there is a single curve that stands out among all the rest of the curves, and that is post-2nd immunization in all 6 cases. So this post-2nd immunization shows a very dramatic shift towards lower mutation frequency in recovered plasmablasts. So that's another fact that we're going to want to examine at the level of individual clones. So at this point, we actually do partition the total set of sequences into clones. And the way that we do this is to develop statistical methods to allow us to do it in principled and not ad hoc ways. And so what we've done is we've developed stochastic models for the 2 processes that are involved in B cell development, or at least the development of BCR. And that is a VDJ rearrangement, which of course is a stochastic process, and evolution by somatic hypermutation, which is also a stochastic process. So in our models, we think of the VDJ rearrangement as assembling the unmutated founder immunoglobulin variable region gene, and then somatic hypermutation to drive it through affinity maturation. And so now that we've got these models, and they're both statistical models as well as dynamic models, in other words, they are generative models for the process, we can now apply them into the definition of our process for determining which sequences belong together in clones. So let's take the definition of clonally related. If two sequences are clonally related, then they have a genetic history such as the one shown in the left-hand panel here. If two observed genes are related, then they are related most recently to an intermediate and originally to an unmutated ancestor. Now, the unmutated ancestor places very severe constraints on the sequences that can be there at that position as at the ancestor. And that actually serves us well, because what we're going to need to do is to sum over all possible ancestors and all possible intermediates. And having done this, we now are in possession of the marginal likelihood, and we could compare that marginal likelihood to the marginal likelihood that we compute for the model on the right. In the model on the right, the observed sequences are not related, and therefore they have two separate ancestors. So in both cases, we're summing over two unknown sequences, but they are very different topologies, if you like. The equation on the bottom illustrates where these various pieces come from and where they go in the equation. So having partitioned our sequences into clones, I want to go through a broad look at how these clones develop, how they affinity mature, and how they compete. So there's a lot of information in this slide, and I will take you through step by step. So each of these rows represents a single clone, and the symbols are placed underneath the appropriate vaccine. So we have pre-immune and then vaccines 1 through 5. The size of this symbol represents the relative size of that clone for that time. So it's compared, for example, this large symbol here occurs at immunization 3, which is to say 1 week after immunization 3. And it shows that clone was very large compared to other clones at the same day. And so among all clones observed after immunization 3, it made up the largest proportion. And that is shown by the size of that symbol. The color indicates the somatic mutation frequency. And in particular, in this case, again, it's the heavy chain that we're looking at. So this top clone is first observed at very low mutation frequency after the second immunizations. No members of the clone were observed either pre-immune or after the first immunization. Between the second and third, the clone grows substantially, at least relative to the other clones, and increases its mutation frequency. Beyond the third immunization, at the fourth and fifth, the clone, relatively speaking, declines in size. Now, we know that all of the clones are declining in size because of that little dip in the total number of plasmablasts recovered, but this is a relative measurement, so even relative to the other clones, it has decreased in size. It persists out to the fifth immunization. It has increased its mutation frequency now by a substantial amount, yet it has not grown any further. you can see that quite a few clones in this subject. This is subject 5, and these are the clones with the greatest single sample abundances. See, a lot of other clones have very similar kinds of patterns of growth and mutation. And in fact, this is sort of what we expect from the textbooks. We expect that there will be elicitation from the naive pool, and that these will continue to evolve, and those that gain a selective advantage by having a higher affinity will continue to grow until they dominate. Now, it's this last piece that we don't see any evidence of here. We don't see these individual clones growing to dominance. We see them increasing and then decreasing. I will point out that there is another pattern of clonal development that we see in all the subjects, and that is clones that we recover even pre-immunization, and they appear at high mutation frequency, and they appear as large, relatively speaking, components of the total plasmablast population. And they are not seen after the real specific response kicks in. We have shown that these clones are not specific for protective antigen, and in fact, we have found that they are autoreactive. So they are not elicited by the vaccine because they appear prior to vaccination. We don't know exactly what these are, but we are working very hard at this moment to learn more about them, and I won't say anything more about them in this talk. We see the same kinds of patterns in all of the subjects. Here are just two more, S1, S2, but the same is found in all of them. Now, we're going to look at the heavy chain mutation frequency for each clone on the day that that clone is first observed. So let's go back to this original clone here. It was observed at 4 different time points. But for the graph that I'm about to show you, we would only count its mutation frequency after immunization number 2. That is the time that it was first observed, and the other observations won't go into the construction of this set of histograms. This set of histograms shows the frequency of cells, or what's the same thing in this condition, antibody heavy chains, as a function of mutation frequency. And so you can see that in the pre-immune and post-first immunization cells, plasmablasts, there's a wide distribution of mutation frequencies from relatively low, but you notice that there's a conspicuous absence of actually very low or naive type cells. And it's not until immunization number 2 that a very large bolus, if you like, of naive cells enters into the plasmablast response. And from that point on, we see very little continuing recruitment from the naive cells, or at least that is visible in these plots. And the mean mutation frequency, or the center of these histograms, is gradually moving to the right. So let's look at an overview of the clonal dynamics in terms of their somatic mutation frequencies. Each one of these bars is divided into high mutation frequency, medium mutation frequency, and low mutation frequency. The total number of these shows you how many of those cells are new at each of these immunizations. So after the first immunization, we see that it's about 50-50 between highly mutated and moderately mutated. At the second immunization, you see that the proportion of highly mutated has decreased, and there's a huge increase in the number of low mutation frequency. And this corresponds exactly to our observation of the influx of low mutation frequency cells after the second from the histograms. The other thing you notice is that the total proportion of cells at this time is almost entirely from new clones. It's the distance between the top of the bar and the top of the chart that gives you the proportion of clones that were previously observed. So it's almost entirely new after the second immunization. After the third, there are more cells that persist from the second immunization, but that number remains relatively moderate at about 50%. So about 50% of the responding plasmablasts are coming from previously unseen clones. Now, we're not seeing a lot of additional naive cells here, so these must be coming from clones that existed at much smaller sizes in previous samples and are now growing. And the same thing holds, at least qualitatively, for all 4 of the subjects who continued throughout the entire study. Well, now let's look at interclonal diversity and intraclonal diversity, because what we're seeing so far is a little bit different from what we are led to believe. So for intraclonal diversity, what we do is we take the clone and compute the mean pairwise nucleotide distance within that clone. Now, the first 2 points are pre-immune, or at least pre the time that the PA-specific clones arise. And at this point, when they do arise, you see that there's a net increase in all cases in intraclonal diversity. And so we would ordinarily take that as evidence of relatively little selection, because if there were strong selection, we would expect that individual lineages, or sublineages, if you like, would come to dominate the clone. But we see that that's not the case. Similarly, if we look at interclonal diversity, we see that after the third immunization, when the clones are all essentially there and competing, there's again an increase in interclonal diversity. This initial decline is mixed up with the existence of these anomalous clones that I mentioned before. But we don't see any further evidence of interclonal competition. Here's another way of looking at the same story. These are plots in which the horizontal stripes, or roughly horizontal stripes, each represents a clone. And the width of the stripe tells you the size of that clone at that period. So what's evident in looking at this plot is that no clone ever comes to dominate for more than one sample period at a time. So this clone, for example, it grew tremendously and became quite a substantial proportion of the total plasmablasts at this point, the third immunization, but immediately decreases in size. And you see that there are no exceptions to that rule. So to take an even closer look at what's going on, I think it's very helpful to actually look at the phylograms that we've constructed using maximum likelihood methods. And so this is a phylogram for the largest clone in subject 1. And the colors of the taxon identifiers give the mutation, no, I beg your pardon, they give the immunization number after which they were isolated. And so very early on, after the second immunization, we see that the sequences cluster very closely together. There are lots of identical sequences. When a bunch of sequences line up right flush with a single vertical bar, it means that they're identical throughout the heavy and light chains. Now, they're also very, very close to the unmutated common ancestor. Now, after the third immunization, they've increased their mean mutation frequency by about 3%. And more importantly, each of these sequences in cyan traces itself back to one of these observed sequences. So these are directly descended from these guys, means that we're capturing a fairly good representation of this clone. But also, you notice that there's tremendous diversification. And so there is diversification of about equal size from all of the precursors. Again, there seems to be little selection going on. After the fourth, there's more diversification, and even after the fifth, now the mutation frequency is up to 7%, which is quite high. And there are still many sequences in this clone that vary from each other by a substantial amount, up to about 14%. And that's not what we expect under the very harsh conditions of affinity maturation that we've come to expect. So you might say, well, maybe affinity maturation isn't really going on at all. Maybe it's just not working. And that's not true. We've synthesized a large number of the sequences that we isolated, and we conducted surface plasmon resonance kinetic analyses to get the off and on rates as well as the equilibrium dissociation constant. Now, in this particular clone, the unmutated common ancestor was actually observed, and that had a dissociation constant on the order of 1,500 to 1,000 nanomolar. After 3 amino acids only were changed, we got about a 20-fold increase of affinity down to about 55 nanomolar. And with a handful of additional mutations, we got down to another 10-fold and at 5 nanomolar. And then it continued to evolve, as you can see here. In fact, the great majority of the evolution occurs between this point and the terminal points. But over that, there was, again, just about another 5-fold decrease, so that the really fast increases in affinity happen early on in the clone, yet the clone continues to evolve, but it doesn't achieve much greater affinity, or I should say that the rate of affinity increase is substantially decreased. And it's not just in this clone, it's in this clone as well, where we see exactly the same kinds of patterns, a rapid increase at the beginning and diminishing returns as the clone ages. So we thought it would be very useful if we could actually assess selection pressure by looking at the nucleotide sequences only. These surface plasmon resonance experiments are expensive and time-consuming, and although there's no replacement for them, you can increase your ability to infer patterns by doing these kinds of selection analyses over nucleotides and then checking back against SPR every once in a while. So we're going to study the selection intensity as the clone grows, and we're going to use techniques that are very common to all kinds of molecular evolution studies. So every nucleotide change either changes the amino acid and makes it subject to selection, or it doesn't, in which case, to a very good approximation, it is not subject to selection. In immunoglobulins, there are two distinct regions with distinct functions. The complementary to determining regions encode the parts of the antibody molecule that bind to the antigen, and the framework regions are responsible for giving the protein the structure it needs to fold properly and to remain stable. And so the expectation is that mutations in the CDR, replacement mutations in the CDR, are more tolerated than replacement mutations in the framework. And in fact, this is so true that evolution has played with codon bias differentially between the CDR and framework regions, so that it's actually easier, or I should say the mutation rate for replacement mutations is higher in the CDR than in the framework. We'll have to take consideration of that effect as well. So what we do is taking that kind of regional bias into consideration, or in fact what we're doing is we're taking bias at the level of the individual nucleotide into account. We compute the probability that a given mutation would fall into one of these two-way classes. So it's either going to be framework silent, framework replacement, CDR silent, CDR replacement, and we can then build up a sense of where we expect the mutations to fall in the absence of selection. Any departure from these probabilities in the observed sequences will provide evidence that there is something going on by way of selection. So to illustrate how we do this in practice and with respect to time, every branch represents an accumulation of mutations from the leftmost sequence and the rightmost sequence. So this is a branch, and in this particular branch, we've observed 5 mutations. And the 5 mutations that we observe happen to be all replacement mutations in the CDR, 5 of them when the expected number was 1.3. None were observed in any of the other classifications. And so we compute this selection parameter, which I'll explain in just a moment, and tie that to the midpoint of the branch. And so we say that the selection pressure at this time is given by the selection coefficients that we've used, that we've computed. Similarly on this branch, and by the way, we actually compute the expected number of mutations based on the actual sequence at the node, the starting position here. So this branch held substantially more mutations, and you can see that 3 of them were replacement mutations in CDR when the expectation was 7.3, and so forth. And in order to actually turn these numbers into useful inferences, we have to have a model for how selection is going to affect the probabilities that we observe. And so we start with a simple multinomial model with 4 categories, and those are the 4 categories that we've already mentioned. And we can, this is just to illustrate that it's straightforward to write out the likelihood. And we can now specify these probabilities in terms of the unselected probabilities, which are designating pi here, times a selection coefficient. Selection coefficient is greater than 1, indicates an increase in the probability. If it's less than 1, a decrease. And finally, because we have a lot of sequences, a lot of clones that evolve over time, we can actually look at the time dependence of the selection coefficients. And what we do is we put all of this together into a big model. It's, not such a big model really. And there are only 4 parameters, alpha and beta for CDR and for frameworks. And once we've ground through all of that, we can actually find the appropriate model. We can find the model that fits the data the best. And in this case, the model that fits the data the best has an increased CDR replacement probability, or what's the same thing, a positive greater than 1 selection coefficient for replacement mutations in the CDR, which is to say even above the codon bias that has increased the probability of getting replacement mutations in CDR, there's additional selection for these mutations. But that over time, this strong selection for novelty, if you like, decreases. The blue line shows where these dots would cluster around if there were no selection at all. Each of these dots represents a single branch, and the size of the spot indicates the total number of mutations observed on that branch. Can also look at the replacement selection in framework, and this shows that there is a decreased selection for replacement mutations in the framework. And again, that's what we expect. What we don't necessarily expect is that negative selection is apparently decreasing over time as well. Now, the reason I say apparently is that if I have strong selection for replacement mutations in CDR, something has to give, because you can't do selection on non-synonymous. And so that means that the total number of replacement mutations in the framework region will apparently be smaller, and similarly, when one decreases, the other increases, unless there's a careful balancing of the time-dependent selection coefficients. See the same patterns in the light chains as well. So at this point, I'm going to stop and tell you what I take away from these studies. So first, the mere statement of fact, the immunogen-specific immune response, at least that registering in the IgG-positive plasmablast population, appears 1 week after the second immunization, not after the first. There is very effective affinity maturation in the clones that we have studied, but in spite of that, intraclonal diversity remains high throughout. So we are achieving high affinity without diminishing the total intraclonal diversity. Perhaps related to that, no clone dominates for 2 or more samples, far from the expectation that we would have memory clones that would continue to grow until they completely dominated the population. We see nothing like that. We can also analyze the temporal trends in selection, and they agree, at least they agree conceptually, with the results of the SPR analysis. And that is that there is selection for novelty, if you like, by which I mean selection for replacement mutations in the CDR. Sometimes this is called positive selection. But that the force of this positive selection, or intensity for this positive selection, decreases over the lifetime of the clone. Now, this is perhaps not surprising. You can think of this as trying to find the top of a mountain by taking steps up, down, sideways. Of course, you don't know which direction the steps are. But once you get to the top, many fewer of the steps are going to take you any closer to the summit. And this is particularly true in the very high dimensions of the genotype landscape. So questions now remain: are these conclusions generic, or are they only specific to this vaccine, to AVA? And we'll have to wait for the carrying out of additional studies on other vaccines, and we are in the midst of doing those now. And now, of particular importance for the HIV vaccine world, we've seen that the intensity of selection decreases over time when the same immunogen is used. Here's an important question: can we keep selection intensity high if we immunize with different immunogens over the time period of the protocol? I think that may well be true, but it'll have to wait until those studies are done for us to know with certainty. So I want to thank everyone who was involved in this study, and particularly to the volunteers who came in and week after week, month after month, got needle sticks. And in fact, they each got 20 needle sticks in all to help us understand how vaccines work. And I want to thank the members of the laboratory, Feng, Kate, Scylla, and Ashin, for all the hard work they put into these studies. And I want to thank Garnet, Akiko, and Masayuki from Garnet Kelso's lab for their participation, and Chip Walter, who directed the clinical part of the study. And of course, I want to thank the NIAID, who provided the resources to make this study possible. Thank you.